Okay. Darken the room a little bit so you can see the screen a little bit better. We're going to pick up where we left off last time, and we're looking at the relationship between amino acid sequence and structure. So the sequence here is a little bit difficult to see. It's highlighted here. And this is a coil-coil. This is the same sequence that I showed you at the end of last time. And what I want to highlight is those non-polar amino acids that are spaced every 3 and then every 4 amino acids apart. Let's see, nope, that one. So I'll highlight those with maybe an orange color. That's not orange. It's pink, okay. So you can see that as we look down the helix, and let's look at it from from the end terminus of the helix down, and we can sort of cut away and see this regular repeating pattern of amino acids that are nonpolar. And you'll notice that the amino acids that are nonpolar are packing into the interface between the two helices, and a nonpolar amino acid on one of the two helices is packed against a nonpolar amino acid on the other helix. So for example, this amino acid here happens to be leucine 5, and it packs against leucine 5 on the other helix. Now, we've given this repeating pattern of amino acids sort of letters, not one-letter codes, but let's see how to say this. So here is leucine, and then 1, 2, 3, 4 amino acids later, one turned down on the helix, you have valine, let's see, that's 10, 11, 10, 9, okay? And then we count another 1, 2, 3, 4 amino acids beyond that, and you have another leucine, and so on. Although that's not the right one. There we go. So, and as we said, because of the repeating pattern of an alpha helix, 3.5 amino acids per turn, because we have this sort of 7 residue repeating sequence where the first and then the 4th amino acid within that 7 residue sequence are nonpolar, that means every couple turns of the helix we're back to where we started. And so, for example, this, if we zoom in on so what we're looking at here is methionine 2, which occupies an A position in what's called this 7 residue repeating sequence called a heptad, and that sequence defines coil-coil structure. And you can see an A position methionine on one helix packing against an A position methionine on the other helix. And let's make this, I don't know, yellow instead of orange. Okay. And then one turn down on the helix, if we zoom in, there we go. Nope, not that. That was the right one. Sorry. One turn down, you're at leucine 5, which on one helix is packing against leucine 5 on the other helix. And that leucine occupies a D position. Okay, so on one turn of the helix, we have an A position nonpolar residue That's methionine. Then one turn down, we have a D position, leucine. And then another turn down, we're back to an A position. In this case, it's valine. One turn down, we're at a D position, which is leucine. A position. Now, you'll notice that actually this amino acid is polar, and that polar amino acid plays a key role that is important, but we won't talk about today. Then a D position, leucine, an A position, valine, and so on. You see these alternating layers of A and D position amino acids within this heptad repeating sequence, okay? And the driving force for this structure to form is the fact that these non-polar amino acids, which would in the unfolded molecule be exposed to water, are no longer exposed to water. They're sequestered from solvent, okay? Now, I've been learning about coil since I started my PhD in 2003, so I no longer can tell whether anything I say about the subject makes sense. So please, let's pause and have you ask questions. Anything you want to know about this structure? Yes?
So the nonpolar ends are facing inward just so they can stay away from water?
That's right. It's called the hydrophobic effect. The nonpolar amino acids are pointing inward. They fit against each other almost like Legos, but the major driving force is for them to be outside of water. Okay, yeah?
So just think of all this, what is it that you want us to take away from this?
This is an example. I understand the question, which is how am I going to see this on a test in some ways? What I really want you to take away from this in life is the relationship between sequence, structure, and function. There are some folks who get the idea from various other sources that protein folding is so complicated that you cannot understand it. And that's simply not true, especially in a case like this where the linear sequence and the patterning of nonpolar residues drives a very specific structure to form. Okay, what else? Let's see, there's some other things we could get from this. We've talked about the nonpolar amino acids that are at the interface between helices. I'm going to just color everything the same, except we're keeping the atoms colored. Yellow atom is sulfur, red is oxygen, blue is nitrogen. What I want to do now is I'm going to color the polar amino acids, particularly the charged amino acids. Let's see, the negatively charged ones, let's go ahead and color red maybe. Okay, magenta since that's easy. Let's do that on both helices. So I'm looking for the E's and the D's, which are the negatively charged ones. Did I miss any? I don't know if you can see the sequence that well. And then let's do the charged ones, the positively charged ones. Let's do blue. Okay, can you see any? There's an R that I missed. Anything else? H, which is sort of physiological pH, maybe only 10% positively charged. I think, nope, there's the R I missed again. We'll color that blue. All right, now we're going to look at the helix from the side. I want you to notice that there are, oops, let's just center it now, kind of off center. I want you to look for times when positively charged side chains are close to negatively charged side chains. One such example is right here. All right, so that's in magenta, that's a glutamate on one helix and a lysine on the other helix, negative and plus. These data, by the way, are from an X-ray crystallography experiment, but they suggest, and you can actually measure the distance between those two helices, or between those two amino acids. and it looks to be about 4 angstroms. That's almost close enough to be touching, so for the atoms to be touching. So likely that's what we call a salt bridge interaction. That is an interaction between a negative charge, an ionic interaction between a negative charge and a positive charge. And you see this at various positions all the way down the helix. There's another salt bridge pair, we might call it, of glutamic acid and a lysine on opposite helices but adjacent to each other. These charged residues that sort of interact with each other across the interface of the helix, they occupy a different characteristic position. Notice they're not buried at the interface between the 2 helices. They sort of flank that space. And that can provide some stability to the helix. It can also be used to control, in this case, what we're looking at is a homodimer, that is one copy of a peptide packing against another the same peptide, just a second copy of it. That's a homodimer, but you could envision heterodimers where 2 different helices pack against each other. And you can sometimes take advantage of complementary charge interactions between lysine and glutamate residues all down the helix. All right. So this is a case where the sequence of the peptide leads directly to its structure and ultimately its function. This is the dimerization domain of a transcription factor. And you need this portion of I'm not showing you the rest of the portion of the molecule, but you need these 2 helices to stick to each other so that the DNA binding domain can wrap around DNA and do what it's supposed to do. Okay. There's, believe it or not, there's more there. There's like hours more we could talk about, but probably we won't. You have hours. That's right. We could just do nothing else. That would be great. There we go. All right. Is that still visible in the light that we have? We talked about that, blah, blah, blah. There's a lot more, but so be it. Okay. So the story I'm going to tell you now is about a class of molecules called antimicrobial host defense peptides. It's part of sort of innate immunity. You have a whole adaptive immune system that can encounter foreign things, bacteria, and so on, and evolve antibodies to recognize those bacteria. But eukaryotic organisms also have just some sort of background mechanism that generally helps kill bacteria. And these are called antimicrobial peptides. The molecule we'll be talking about is called meganin-2, and it is from frogs. The name meganin is actually from a Hebrew word that means shield. So it's a molecule that acts as a shield for frogs, secreted from their skin. It has antimicrobial properties. And so we're going to talk about why it's antimicrobial. This is the sequence. It's 20 or so amino acids long, and in green I've highlighted all of the non-polar ones. And then we're mapping this sequence onto an alpha helical conformation. This is called a helical a helical wheel diagram. It basically, you start at, I can't even find amino acid 1, and you go around in a pattern that's 3 and 3.6 residues per turn. And so this shows you sort of a view from the N-terminus of the peptide of the overall characteristics of this sort of helix that we're looking down. Think of it as a cylinder, and along one face of the cylinder, because of the positioning of these nonpolar amino acids, one face of the cylinder is more nonpolar than the other. So in green, as I said, are the nonpolar ones. In orange, I think, are the polar or smaller but neutral ones, and in blue in particular, or gray, whatever that is, are charged amino acids, some lysine amino acids, for example. So and if you sort of relax your eyes a little bit, the overall pattern you get is one face of the helix is nonpolar, and then the other face is positively charged. And that overall characteristic is the defining characteristic of antimicrobial peptides. There's a lot more of these. Some of them are more selective for bacteria than for other things. Some of them actually start to cause problems with red blood cells as well. There's an antimicrobial peptide called melittin that's from bees. It's in bee venom, and it causes pain associated with bee stings because it does to your cells the same thing it does to bacterial cells. All right, so in order to understand its mechanism of action, you have to know a little bit about phospholipids and the phospholipid bilayer. As you've learned about the phospholipid bilayer before, particularly in bacteria, the polar head groups are negatively charged. A phospholipid might look something like this. Where these are long chain fatty acids. So the bacterial cell membrane is predominantly negatively charged, whereas the peptide that we're talking about has a nonpolar face and is positively charged. And it's thought that that positively charged face sort of sits down on the polar head groups of the bacterial cell membrane as shown here, whereas its nonpolar face sort of intercalates into and packs against the nonpolar surface of the interior of the lipid bilayer. And you get enough of these in a localized area and you blow a hole in the bacterial membrane. And bacteria are jam-packed with like DNA and proteins. You blow a hole in the membrane and it's like popping a balloon. Their sort of guts spill out into everywhere and they're dead. So this is a general strategy / mechanism for inhibiting bacterial growth is with antimicrobial compounds, and they generally have the characteristic that meganin does. That is, they are globally what we'll call amphipathic. That means they have two, or amphiphilic is fine too. They have one side of them that's hydrophobic, another side that's hydrophilic, positively charged, and that gives them their function. Dr. Savage in the chemistry department here has a very successful long-term project inspired by antimicrobial peptides. He's actually developed some smaller molecules that are not peptides, but they share this general characteristic of having a positively charged face and a non-polar face. And they're actually really good at inhibiting bacterial growth. He's using these now to coat, he's coating medical devices with some of these molecules to inhibit growth of bacteria, say, in endotracheal tubes and stents to prevent secondary infections in hospital settings. Okay, so that's all of that. Anything you want to ask about antimicrobial peptides? Yeah. Okay, go ahead. Sorry. Yeah, shoot.
Why doesn't that inhibit cells of eukaryotic cells?
Why doesn't that inhibit eukaryotic cells? Good. Phospholipid head groups are different in eukaryotic cells. So, as I pointed out, and I have to draw the phospholipid again, here's the long nonpolar tail. For bacteria, often this is just a negative charge. In eukaryotic cells, that's often other things. For example, in phosphatidylcholine, it's that, which is neutral. So that's the origin of the selectivity. Eukaryotic cells are not so universally negatively charged. Yep. Often a measure of how good one of these is going to be as an antimicrobial peptide is you compare the concentration at which it kills bacteria versus the concentration at which it causes hemolysis, that is, makes your red blood cells burst open. And naturally, you want that concentration to be really low. Okay, yeah. Same question? Okay, cool. What else? Okay, so let's talk solid phase peptide, or let's talk about peptide synthesis. Your basic problem in making peptides or proteins is a problem you encountered before in one of the early chapters about carbonyl chemistry. And that is, if you want to make an amide bond using a carboxylic acid and an amine, what you must remember is that you generally can't do this directly, right? It looks lovely on paper. Oh, it's just a dehydration reaction. We have the nitrogen attack the carbonyl carbon, we get a tetrahedral intermediate, perhaps some proton swapping, and the leaving group leaves, and we have our new amide bond. Sounds good, but in practice what happens is you have this acid here, which has a pKa around 4, and amines are bases. And it's actually the pKa of the conjugate acid of the amine, the ammonium ion, actually has a pKa of around 11, although it's closer to 9 for amino acids. But the point is that before you even get to make the amide bond or attack the carbonyl carbon, you just do acid-base chemistry to generate the carboxylate conjugate base and the ammonium conjugate acid. And then you're congratulations, you made an ammonium salt. Whoop-de-doo. It's not an amide bond. And so our strategy for making amide bonds, as you've seen, has been to turn the OH into a good leaving group first, and then have the nitrogen attack. So that's problem one, which we have solved, right? We use, to make an amide bond, we have, our solution is to use DCC, which is dicyclohexyl carbodiimide. That's a reagent you can memorize. We're not going to talk about how it works anymore. We did that back in an earlier chapter. You need a base to soak up this proton on the carboxylic acid. Basically, DCC converts the OH into a really good leaving group. In a second step, you add your amine, and that makes the amide bond. Okay? So that's review from, and I no longer remember which chapter it was. It was in the range of before 16. 16, 15, something like that. Okay? Now, that's all well and good, but we're making amide bonds involving amino acids. And so we've got a selectivity problem here, because the molecule that has your carboxylic acid in it also comes preloaded with an amino group. So what if I wanted to make an amide bond between this alanine and, let's say, this leucine? What's the problem with just using this same approach? What's the problem of just using DCC and then putting these two things together? Both of them have carboxylic acids. Both of them have amines. So if we try if we just add DCC and a base, we run the risk of getting, and this is alanine and this is leucine. I'm going to draw the desired alanine leucine dipeptide. We'll call that Al. But we run the risk of getting La and LL and AA as well, right? I'm not going to draw all those structures, but do you see you've basically got two different carboxylic acids and two different amines. And so you're going to get all the possible combinations. Okay, that's a problem. So we need a way to solve that problem. Nature solved it with the ribosome and the genetic code. Humans have solved it using protecting groups. And in particular, the protecting groups we use are for the amino group, for the alpha amino group, okay? There's a couple of different options. I guess they're both for the amine and for the acid. So let me show you a couple of options for the protecting groups for the amine. The first is called Bach. And I'll show you what it looks like. Remember, what makes a good protecting group good is that it stops whatever functional group you're interested in, stops it from being a problem, but it has to be easy to put on and easy to get off. So what I'm showing you here is Bach protected alanine. This group is called Bach. That stands for, actually it used to be called T-Bach, but only overachievers call it T-Bach. And the B stands for butyl, O stands for oxy, and C stands for carbonyl. T-butyl oxycarbonyl. The functional group is a new one, a carbonyl bonded to an oxygen and a nitrogen. That functional group is called a carbamate. And carbamates are very common in protecting groups. Let's talk about why. So the three things you need for a protecting group is it needs to attenuate the reactivity of an offending of the offending functional group. So we need to attenuate the reactivity of the amine. How it does that is by tying up the lone pairs on the nitrogen in resonance. So they're no longer nucleophilic. Okay? So that's criteria 1. 2, it has to be easy to put on, and it also has to be easy to remove. So let's talk about how you first remove the Bach protecting group. It turns out you can remove the Bach protecting group under acidic conditions. And maybe we'll say strongly acidic conditions. Basically, the mechanism is it's SN1-like, and it involves a decarboxylation. As you might expect, by virtue of being irreversible, decarboxylations are high-yielding reactions that really help things move forward. And they are very common in protecting group removal type reactions. So what happens under acidic conditions? I'll sort of draw the mechanism. Under acidic conditions, you can and you need a strong acid. This can be HCl or it can be trifluoroacetic acid. Under strongly acidic conditions, you protonate the nitrogen, and then the carboxylate group, I'll show this stepwise so you can show it in a concerted fashion and it's fine. The T-butyl group gets left behind as a carbocation. What you're left with then is this, I guess I'm showing you one of the possible mechanisms. This intermediate, which can do a decarboxylation reaction to give you the neutral amino acid. What happens to the T-butyl carbocation? Generally, you're doing this in water with a small amount of water present, so that probably goes on to form T-butanol, which you can remove. It's very easy to separate that from your free amino group. All right. Putting the Bach group on would simply involve, so let's see, this is what I just showed you here is the deprotection reaction. The protection reaction involving Bach would be to take an amino acid you've actually seen this previously on an exam. You take a reagent called Bach anhydride. People just prefer to write this like Bach 2O because it's cute and abbreviated, but Bach anhydride, and we're out of space. Looks like this. The protection reaction basically involves a nucleophile attacking the carbonyl carbon, getting a tetrahedral intermediate, and then the leaving group leaves. We don't need to go into that in a huge amount of detail, but this is just basically nucleophilic acyl substitution. And then people will abbreviate Bach with just the 3 letters BOC. I'm getting stereochemistry funny, sorry. Even though what they mean is that, okay? All right, so how would you use this as an example if you wanted to make an amide bond? Let's have, well, actually, before we can do that, I need to show you a protecting group for the carboxylic acid. But anything you want to ask about the Bach protecting group for a nitrogen? Okay. For the carboxylic acid, we're going to use what's called the benzyl ester. There are a lot of other things you could use. This one happens to be really convenient, easy to do. What is the benzyl ester? Well, let me draw a benzyl ester protected carboxylic acid. You have a CH2 group and then a benzene ring. You can make this, so the benzyl ester, let's talk about how it attenuates the reactivity of the carboxylic acid. The benzyl ester is not a very good electrophile. It's tough for a nucleophile to come in and attack the carbonyl carbon, meaning that once the benzyl ester is on, the carboxylic acid is no longer reactive to amines. Okay? Part of that may be steric hindrance. Part of that may also be that the oxygen is not a particularly good leaving group. Installation of that benzyl ester is easy. You do this with in an SN2 reaction with benzyl bromide or benzyl chloride and your carboxylic acid. Sorry. Struggling here. You usually need a non-nucleophilic amine base to get the carboxylate, and then that is nucleophilic. Okay, so that reaction is easy, and removal, notice this is different from ester hydrolysis, what's going on, or ester formation under acidic conditions. We're making the conjugate base of the carboxylate, and we're doing an SN2 style reaction, all right? Deprotection is also easy. You could do ester hydrolysis, but actually it's easier to do what's called hydrogenolysis. You maybe remember learning that a benzyl radical is a decently stable kind of radical because the radical stabilized by resonance. For the same reason, it's actually really easy for a palladium catalyst to add hydrogen across this bond between the oxygen and the carbon. So that's called palladium catalyzed hydrogenolysis. You add hydrogen and palladium on carbon. What that does is it breaks the bond between the oxygen and the carbon adjacent to the benzene ring. One hydrogen from H2 ends up there. The other one ends up here. And hydrogenolysis is a really fast and easy reaction to do. There's no byproducts, essentially. I guess you've got toluene as a byproduct, but that's easy to separate from your amino acid. Okay? So, we've got a protecting group for the amine, we got a protecting group for the carboxylic acid. Let's now go back to what we wanted to do, which was make al, and let's show you how you can do that without undesired side reactions. So if my goal is to make al, by the way, we don't normally pronounce peptide sequences. We can call it AL if you want to. Just thought it was funny. All right, so my peptide has an N terminus and it has a C terminus, an amino group and a carboxylic acid group. If I want to make the peptide AL, I need alanine to be the carboxylic acid, but I don't want the amino group of alanine to interfere in the reaction, so I'm going to use Bach-protected alanine. Okay? That means if I use Bach-protected alanine, Bach on the nitrogen of alanine is now masked and it's not a problem. It's not going to interfere with the reaction. Similarly, if I want leucine to be my C-terminal amino acid, I need the amino group of leucine to be free, but I need the carboxylic acid of leucine to be protected, and you can do that with the benzyl ester. I should have pointed out that out of laziness, we often abbreviate the benzyl ester with an O and a B and an N, OBN. Okay? With those two protecting groups in place, I'm fine now to use DCC and triethylamine to make the amide bond. And remember, DCC activates this carboxylic acid, doesn't touch the benzyl ester. Then the nitrogen comes in and attacks the carbonyl carbon. You get a tetrahedral intermediate, the leaving group leaves. Then I've made my amide bond between the carboxylic acid group of alanine and the amino group of leucine. I've got the right connectivity. I don't have any of the undesired AA or LL or LA products. Now my only problem is my final goal was the dipeptide, but I now have these protecting groups on there. So what do? Well, I can remove them. Pretty straightforward. It probably doesn't matter which order I do this in. If I wanted to remove the benzyl ester, I would use hydrogen and palladium on carbon. And then if I wanted to use the, remove the Bach group, I could use a strong acid. Trifluoroacetic acid is a common one. It's called TFA. All right. And that would give me my desired product. All right. Yeah.
In the book, when it was adding the carboxylic acid protecting group, it used phenol in an acidic solution.
It used what?
It used phenol in an acidic solution. Does it matter which one you want?
Oh.
Or
Probably, I'm just checking, probably it didn't use phenol because phenol is different from benzyl alcohol. Phenol's like this and benzyl alcohol's like this. This would be too much of a good leaving group and might be trouble. Yes, you can make that benzyl ester if you wanted to. There's you could also have done that using benzyl alcohol. Sorry. So I'll just abbreviate that here. A carboxylic acid, benzyl alcohol, and acid catalyst. This is basically Fischer esterification conditions. And that works just as well. Naturally, if you wanted to benzyl protect an alcohol that already had a Bach group on it, acidic conditions is a terrible idea because you're going to deprotect your nitrogen. Yep. All right. So what I just showed you is basically solution phase peptide synthesis. And we might continue onward. If suppose I wanted to make something else, right? Suppose I didn't want a dipeptide, I wanted a tripeptide. You can see how I could have at this stage said, okay, let's actually remove the acid protecting group. Let's leave the nitrogen protecting group on. And then I can bring in another amino acid and add it to the C terminus. We can pick whatever we want now. Maybe make phenylalanine. We would use the benzyl protected phenylalanine but with the free nitrogen. And then we would use our Bach protected dipeptide but with the free carboxylic acid. Then we can use DCC and triethylamine again, and that's going to make our next amide bond. And sorry, I'll try to clean up the penmanship here. I think I'm hustling too fast. NH is the new bond. Okay. Now we've got a tripeptide, ALF, which that reminds me of a short-lived show in the 1980s with the alien in it. It was kind of interesting. You should Google it. But so I could continue, and here I've added another amino acid to the C terminus of my peptide. However, I could have also come here and instead of benzyl deprotecting, I could have Bach deprotected and then added another amino acid to the N terminus. So I've got some flexibility. Now, there's some practical reasons why you'd want to do one versus the other that we don't need to get into here. But one thing I want to point out is how many reactions we had to do to get to this point, okay? We had to do a protection reaction on that amino acid. So that's one. We had to do a benzyl ester protecting reaction on this amino acid, so that's two. We did the coupling. This is what we call coupling of two amino acids together under amide bond forming conditions. That's three. Then we did another, this is now a deprotection reaction. That's four. We had to do a protection reaction on phenylalanine, that's five. A coupling reaction, another coupling reaction, that's six. And then if we wanted to get the tripeptide that's deprotected, that would be 7 and 8, okay? 7 or 8 steps. You better hope each of these is about 100% yield because you're gonna take a hit every time, and overall yield is multiplicative. That is, if it's 70% yield in one step, it's 0.7 to the 7 for 7 steps, and that number starts to get small. We can figure out what it is. I can't do that in my head. Calculator. So yeah, 0.7 times 0.7. That's 2, 3, 4, 5, 6, 7. For a 7-step reaction, 70% yield gets you overall 8% yield. Not great, right? In contrast, if you got 99% yield and you do 7 steps, let's see, 2, 3, 4, 5, 6, 7. 99% yield each step, you got 93% yield overall for a 7-step reaction. So it's really important that the yields be high. There's another practical issue. I want you to think about how you would separate starting material and undesired side products from your desired product. This is a practical issue we don't often ask you to think about, but if you go in the lab, you actually have to do this. So if you're in Professor Castle's lab or Professor Michaelis's lab and you're making a tripeptide, and you do this reaction, at the end, you've got to separate your desired product from unreacted starting material if the yield isn't like 100%. I want you to look at the characteristics of these starting materials and compare them to the characteristics of the tripeptide. Normally, one of the major forms of separating molecules in OChem is called chromatography. You separate them based on polarity. I'm not sure whether you get to do that in 353 or not. I don't remember. If you take the 455 lab, you certainly will, but that's for people that really love OChem. In any case, it can be difficult if you look at the overall polarity. There's not a huge amount of daylight between the polarity of these two starting materials and this product. So separation might be difficult. So in the 60s, chemist Bruce Merrifield at the Rockefeller Institution invented a way to do this that doesn't require any purification in between coupling and deprotection steps. One thing you might, if you needed the reaction to be higher yielding, one thing you can do, a common strategy would be, let's just use an excess of one reagent to drive the reaction to completion. Great idea, but now you made your problem even more difficult because at the end, how am I going to separate my 1 mole of product from my 5 excess moles of starting material? Merrifield found a way to do that, and it revolutionized, that's not a word. Revolutionized, that is the word. Revolutionized is not the word. It revolutionized synthesis of peptides and really enabled chemists to get into the area of peptides and proteins. He invented something called solid phase peptide synthesis, which we'll talk about after the break. kind of a nice. We pause now for a few messages from our sponsors. All right. So, want to spend just a few minutes talking about how solid phase peptide synthesis works. As I said, the person who's responsible for this is named Bruce Merrifield. That's not a test question, but he's a Nobel laureate, won the Nobel Prize in the 1980s for the work that began in the 60s to generate this. And it's why today, if you want, you can go online to any number of peptide synthesis vendors and order a peptide. You type in the sequence, they mail it to you. It takes probably a few days, but the process is pretty quick. We do this routinely in my lab all the time. Solid phase peptide synthesis is based on doing chemistry on a solid as a reaction vessel. So the solid that's the basic reaction vessel for peptide synthesis was polystyrene. And we may say more about polymers. I think we talked a bit about polymers in 351 when we talked about radicals. Polystyrene is, so styrene is just a benzene ring, oops, don't like that, with an adjacent alkene. Polystyrene you get from radical polymerization and of styrene, and it's alternating two carbons. Every other carbon has a benzene ring attached to it. You get the idea. And these are we can highlight in different colors the individual styrene units of this polymer. We don't need to talk about the radical polymerization process. Every so often in polystyrene, you can actually control how, and this is, thousands of units long. In fact, it's so long that we no longer care what the end groups are, because no matter what they are, the polymer is mostly that linear chain of styrene. You can make the polymer more rigid or more flexible depending on the conditions you use. If you use this is called divinyl benzene. That's basically a cross-linker. This is styrene. And what divinyl benzene does is it takes one of these units, one of these benzene rings has two handles on it, right? And so it can be connected to another chain of polystyrene. And I guess you guys can, well, trying to decide how much to try to make a pretty drawing. So anyway, you incorporate divinyl benzene as a cross-linker, and that can change the properties of the polymer, make it more rigid, make it tolerant to different conditions, temperature, and so on. And then the third magic ingredient was a version of styrene that had a leaving group attached to it. Okay, so chloromethylbenzene makes it so that one of these benzene rings, let's choose the pink one, will actually draw out the benzene ring, and it has a leaving group on it. And Merrifield reasoned, hey, why can't I do substitution chemistry there and attach something to the polymer and then do whatever I want and then eventually cleave the polymer from the molecule that I'm working on, okay? So we're going to abbreviate all of this as a circle with a PS in it, and that stands for polystyrene. Often the polymer gets broken up into sort of bead, spherical shaped things. And we're going to remember that one of the things attached to this bead is a chloro-leaving group. Of course, this polystyrene bead would have many such chloro-leaving groups attached to it, okay? So then, Merrifield said, let's do an SN2 reaction to put an amino acid, a Bach-protected amino acid, on the peptide, on the polystyrene bead. All right, that ought to be pretty straightforward. Nucleophile attacks, leaving group leaves. So now here's my amino acid. And at this stage, I should point out that the polystyrene bead is large enough to see, and it's large enough that you can isolate it easily from a solution. You could do that through centrifugation, you could do it through filtration. There are things called Fritted filters that allow liquid to pass through but stop larger things. And this bead is large enough, the polystyrene bead is large enough that you can do conceptually what you would always like to do, which is reach into the reaction mixture and pick up the desired molecule and leave everything else behind, okay? So once you've loaded this amino acid onto the polymer, which we call resin, I don't know why we call it that, but that's what we call it, then you can do chemistry. So for example, I can use TFA, trifluoroacetic acid, TFA alone is enough. There's usually a little bit of water there. I can use trifluoroacetic acid to deprotect my amino acid, thereby unleashing the amino group. So that's a deprotection step. And then in a second step, I can do a coupling reaction where I use DCC, triethylamine, and then my next amino acid, Bach protected so as to avoid the problems we've talked about before. But this time, because I can physically isolate my resin, I can use excess of my coupling reagents, maybe 5 equivalents, 3 to 5 equivalents. That means for every 1 mole of amino acid I have on my resin, I have 5 moles of my next amino acid that's activated. And this promotes high yield. All right, so I do this in a reaction vessel. And when the reaction's done, what I have is my desired dipeptide on the resin. Here's the new amide bond that I made. But I'm still going to have 4 equivalents of sort of unreacted starting material. And so normally that would be a problem. In this case, though, the reaction vessel is often, it's either a glass or a plastic cylinder. And at the bottom, you have an outlet that has some sort of switch that you can turn to open it up or not. At the bottom of this reaction vessel is a filter. It's called a frit. It has pores in it that allow the liquid out, but not the beads, not the polystyrene beads. We'll just use, so that's those are the polystyrene beads, and then you have your reaction mixture, the solution, your reagents. I don't know why we're coloring it green, because it's not really green, but anyway, you got these resin beads. It's easy at this step, and in green is where your unreacted reagents are. You turn the stopcock at the bottom, and all of the solution comes out, and afterwards what you're left with, you dispose of the undesired, oh shoot, trying to do too many things at once. You dispose of the unreacted starting material, you rinse off the resin beads, and you have in one very easy step purified your compound, and you're ready to go to the next step. So it's just purification by washing resin beads. Okay, very fast. Usually you rinse, I don't know, 3 to 5 times. And that takes you to the next step where you're able to do another deprotection using TFA to remove Bach. And then another coupling reaction with your third amino acid. Oops. DCC and triethylamine again, and that will put, and again, you rinse. After you do the deprotection step, I should say, you rinse out the TFA, and you drain out the TFA. So in between each of these deprotection steps, there's a drain and a rinse. That allows you to isolate your desired product, and so iteratively, we basically string each new amino acid on. I think I'm getting stereochemistry completely wrong here. So, because I'm drawing this in a way that I'm not used to. Oh well. Okay, so it's this repeated iterative pattern of deprotection, coupling, draining and rinsing in between that allows you to put one amino acid after another onto this growing chain. Okay? The process is iterative, meaning a robot can do it, and so we now have automated synthesizers that do this. And we actually have one upstairs that the department shares in space between my lab and the Castle lab, where you literally supply the resin and the amino acids and the coupling reagents, and the robot does everything else for you. You can also do it yourself by hand, and if you do it, a lot of the deprotection reactions are pretty fast. The coupling reactions can be slower. If you're doing this at room temperature, coupling reactions take about an hour long. If you're doing this at elevated temperature in a microwave like we have in my lab, the deprotection steps last a couple minutes. The coupling steps last probably a 15-minute cycle from start to finish of coupling and deprotection. So in a day, you can make a 30 residue peptide if you're paying attention. Now, because it's an iterative process, it's easy to get bored and forget where you are and forget that you've just done a deprotection step when you thought you did a coupling step. So there's lots of things you can do to mess the process up, which is why we like the robots. But if you're paying close attention, I can't tell you how many times I've messed up peptides at various points in my career. But this is how you do it. Now, at the end, suppose you make a 30-mer. At the end, likely you don't, a 30 amino acid peptide, at the end, likely you don't want to have Bach on there. So you do a final deprotection step to remove the N-terminal protecting group. And then you have to have a method of cleaving this bond between your peptide and the resin. Otherwise, you just spend a lot of time and money to make some fancy polystyrene that has some molecule on it, which may have its uses, but we need a way to cleave the peptide from the resin. In the Merrifield's original polymer, cleaving from the resin was done with hydrofluoric acid. Now, HF, as you may know, has a pKa of around 4. It's not particularly acidic, but it is a really dangerous reagent. You use HF actually to etch glass. Its bonds with silicon are pretty strong, so it's good for etching glass. But it can also cause like lethal damage to bones and other biological structures. Like if you work with HF, you have to have a special fume hood to work in. You have to have constant access to a reagent that will protect you if you get any on you. It's just tricky. People do it, and people that do it get used to it and they don't mind it, but you never want to get any HF on you at all or you could die. So naturally, the only people that did this stuff in the beginning were people that had trained in Merrifield's lab and knew how to do it and were used to it. But sometime later, a variation on the Bach protecting group was discovered, along with a variation in how to attach the peptide to the resin that made the chemistry a lot more friendly. So I don't think this is in your text, but I do want to show it to you because for the most part today, in solid phase peptide synthesis, people are no longer using the Bach protecting group and are no longer using this particular kind of resin. So let's talk about this alternative to HF cleaving. Cleaving the peptide from the resin. Sorry, I guess I should have showed you the structure of our peptide, nice and cleaved from the resin. Cleaving from the resin is irreversible. Once it's off the resin, it's not going back on. Okay, and you got your peptide. So an alternative to HF cleaving is to use a resin where we functionalized I'm going to get this wrong, so I need to look it up. Okay, got it. So they modified notice that in the original Merrifield resin, you had your peptide was attached to this benzylic carbon. The innovation was to put another benzene ring in between that benzylic carbon and where you want your peptide. So I'm just using R for your growing peptide chain. It turns out that if you do this, This oxygen at this position in this now linker between the resin and your peptide actually under acidic conditions can undergo an SN1-like reaction. Imagine, protonating the carbonyl oxygen under acidic conditions, and then having this group leave. That would be your peptide leaving in sort of an SN1 type fashion to give you your free peptide. And then the reason this can happen becomes clear if you look at the carbocation that's left behind. Looks like a primary carbocation, right? Wrong. Exam question. Which atoms in the ring share the positive charge? You can draw a resonance structure in which the positive charge on that carbon that's left behind after your peptide leaves is actually on that oxygen. And, you know, here, 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 or here. Okay. So, I think I'm getting that wrong, aren't I? No? Well, in any case, you can delocalize that positive charge because of the resonance donating ability of that attached oxygen. That makes it so the peptide comes off under SN1 conditions, whereas up here, cleaving from the resin actually happens. I didn't show you that mechanism. I'm not going to. It doesn't matter too much. It actually probably happens under SN2 conditions where fluorine acts as the nucleophile and attacks the benzylic carbon. So a minor modification in the structure of the linker made it so that under TFA conditions now, and TFA isn't the same as HF, it's a basically souped up version of vinegar. It's a strong acid, but you can remove now a peptide from resin using a strong acid instead of HF. Now, if you've been following along, TFA is the same thing we're using to remove Bach. So that's bad, right? Because if at this earlier stage, when I go to do a Bach deprotection, if my peptide also falls off resin at that point, that's bad, right? Because then I can't do any subsequent steps. So, having developed a new way to do a cleaving reaction, we also needed an alternative to Bach. We need basically a version of Bach that is not what we will call acid labile. That means does not deprotect under acidic conditions. So if you remember the structure of Bach, I'm just going to use R for the rest of our amino acid. The structure of Bach had this carbamate functional group, and we saw before that when under acidic conditions, you could protonate the nitrogen and then the Bach would leave and the negative charge left behind on the oxygen would do the decarboxylation reaction, okay? The alternative to Bach keeps the carbamate structure, but it alters what's on the oxygen. And in this case, it's not a T-butyl group, it's something that's quite a bit more elaborate. Okay, so this is our new alternative to Bach, and it's called FMOC. Nobody pronounces it FMOC, okay? Nobody says FMOC, they just say FMOC, all right? You can if you want to, it's kind of fun, but nobody will know what you're talking about. Yeah, you walk up into my lab and just try this on my students. Where are your FMOC amino acids? You're like, what are you talking about? So this F stands for this hydrocarbon, time. F stands for that hydrocarbon, which is interestingly called fluorine, but not the kind that's an atom. It's spelled differently. I didn't make this stuff up, I promise. I would have chosen something different, but that's what it's called. FMOC is base labile. So what do we mean by this? FMOC cleaves in sort of an E1CB type mechanism. So this proton is somewhat acidic. Could you tell me why? Why is it somewhat acidic? Resonance stabilized and, aromaticity. How many electrons would be part of that conjugated pi system if we removed the proton?
14.
The whole thing, right? Polycyclic 2, 4, 6, 8, 10, 12, 14 there. Is that an allowed number for aromatic? Okay. So if you can make something that's aromatic by deprotonation, what that means is that proton is more acidic than you'd otherwise expect. So a base can remove this proton. That's kind of cute, right? It's a modified protecting group that uses principles that you have recently learned. You now should know why that proton is acidic. I suppose I could ask you why that proton was acidic in an exam setting, and I wouldn't feel bad about it. Here's the conjugate base. Okay. Now, again, we've said that conjugate base is aromatic. Adjacent to that conjugate base, you have something that maybe doesn't look like a leaving group, but it's sort of a spring-loaded leaving group, because if we make a new pi bond between the carbon of this 5-membered ring and the adjacent carbon, we kick that group off. We now have something that can lose CO2. So I'm showing this to you stepwise, though some people draw it in concerted fashion, and that's fine as well. I'll draw the hydrocarbon that forms when FMOC leaves. This, you don't need to know this. I don't know why I'm telling it to you. This is called dibenzofulvene, and it is UV active. You can actually watch it. If you add to your robot a UV vis spectrophotometer, you can actually watch this come off your sample as you're doing the deprotection reaction. This is often used as a way to determine, that the deprotection reaction actually worked. Then you're left with the conjugate base, or rather the leaving group, which can do a decarboxylation reaction. Probably the nitrogen picks up a proton from some source. I'm not sure what it is. Possibly the conjugate acid of the base that you used as the leaving group, as the decarboxylation is happening. You lose CO2 and you free up your amine. So FMOC now allows you to do basic deprotection reactions. Coupling reactions are exactly the same, but your peptide never sees TFA until the very end when you cleave it from resin. So this is called the FMOC approach to solid phase peptide synthesis. An FMOC synthesis would be exactly the same as a Bach synthesis, only instead of using TFA for the deprotection, you're going to use a strong base. The base of choice is either piperidine, which is a 6-membered ring containing nitrogen, or I think piperazine, which is 2 nitrogens. Either of those work okay, and we've used both in my lab. So I'm not going to draw a whole FMOC solid phase peptide synthesis for you, because it's exactly the same as Bach synthesis, only you switch out the deprotection conditions and you use a different resin. Resins are now commercially available. FMOC protected amino acids, Bach protected amino acids are commercially available. There's more I could tell you because we haven't said anything about side chains. And we've just put R groups here. But remember that some of these side chains themselves might have nucleophiles on them, right? So, though I didn't mention it, for example, lysine, I don't think I got enough carbons in that lysine. There we go. Lysine has an amino group, which itself could interfere with those coupling reactions, right? So you need protecting groups for the lysine side chain. I'm not telling you about what those are. You can learn them later. It's not that hard. But basically, and there's strategies for dealing with it. We haven't said anything about it. But basically, FMOC synthesis is going to be the same as Bach synthesis, only you're using different cleaving, you're using different deprotection conditions to remove FMOC versus Bach, base instead of acid. And then at the end, you're cleaving your peptide from resin using acid instead of HF, which means that average folks like me can learn to do it without having to have a whole bunch of infrastructure. Right. Okay. Anything else you want to know? Yeah.
So, do we use FMOC in basic conditions?
You cannot use FMOC in basic conditions because base removes it.
Okay, so we won't use that in acidic conditions.
So FMOC can tolerate acidic conditions. Your coupling conditions don't affect FMOC or Bach. Right. So remember, the goal of your protecting group is to keep the amine from being reactive until you want it to be. And the reason we have chosen to use FMOC, which is not acid labile, is because we're using strong acid for our deprotection conditions. And if we used strong acid for, we use strong acid for our cleaving conditions. If we had used Bach, anytime we went to do a Bach deprotection, our peptide would have been cleaved from the resin and would have gone away in the wash, right? So in an FMOC synthesis, your peptide never sees acidic conditions until the very end when you cleave it from resin. You only use basic conditions in deprotection reactions when you want to remove FMOC. So I guess I could call this base labile. Sorry. What else? Saw another hand. Go ahead.
Is there a reason we would use the first method instead of this solid phase peptide synthesis?
Yeah, is there a reason you'd use the first method, the solution phase versus solid phase? Yes. Solid phase generally is fairly small scale. So in general, you've got less, 1/2 to maybe 1/4 millimole reactive handles for every gram of resin, which means that on a reasonable scale synthesis that's easy to handle in the lab, we're making milligrams of peptide. If you want to make grams or kilograms of peptide, there are companies that do massive solid phase peptide synthesis in huge reactors. But in the laboratory scale, it's sometimes easier to get larger amounts of peptide via solution phase. The problem with solution phase is, though, that yield, you can't get very long before yield starts to drop off. So there are strategies for this. For example, in Dr. Castle's lab, they've been working on a number of peptide natural products, but one of them is called yacuamide, and it's, I can't remember how long, but they basically plan the synthesis by making segments, like a five amino acid peptide segment here, a 4 amino acid peptide segment here, a 6 amino acid peptide segment here, and then only at a late stage do they link those two together. So that's called segment condensation. It's different from linear synthesis. It has its own challenges, but that's one of the strategies people take when they want to do solution phase. sort of an interesting historical fact, in the 60s, Merrifield used this method to make ribonuclease A, which is a 120 amino acid protein. And it was a real tour de force, like just brute force slogging away getting it at the same, via solid phase peptide synthesis. At the same time, a chemist named Ralph Hirschman, who worked for the company Merck, was competing with him using solution phase synthesis to make the same thing. And they both succeeded and I think published back-to-back papers in Science or Nature or whatever it is. But that was one of the early demonstrations that all you need for some proteins for them to be active is to have the right sequence. Doesn't matter where you get it as long as it's got the right sequence. What else? Okay, so that was all just like the basic orientation to doing work in my lab, actually. And peptide synthesis can be fun and rewarding. We actually are not so interested in the chemistry of making the peptide, but actually understanding the structure and function and folding of the peptides and proteins that we make. All right, so with that, we are ready belatedly to move to the next chapter, which is carbohydrates. So I estimate now we're about a full lecture behind. That's okay because the university generously gave us a Tuesday as a pretend Monday at the end of the semester. Yeah. So we'll see whether we use it or not. I'm guessing you guys vote no, which is fine with me. We're going to talk about carbohydrates next. And my goal was to do carbohydrates, lipids, and nucleic acids before the exam. I don't think there will be that much to do on nucleic acids. We may skip it. I hate to do that, but carbohydrates we'll definitely do. There will be things here in this chapter that you have to memorize. There will be other things that you don't. So we're going to begin with just the basic structure of glucose in its cyclic hemiacetal form. And I'm going to talk about what the various portions of the structure, how they're related to the name glucose. So carbohydrates. Hopefully you're good at drawing a chair and not the kind you can sit on. This is the cyclic hemiacetal form of beta D glucopyranose. Sorry about penmanship. Pyranose. Okay? That's an obnoxious name. Most people will look at that and will call that glucose. Let's talk about what each portion of this name means. First, the ose tells you that this is a carbohydrate. And all carbohydrates should have names that end in ose. So you've heard of ribose before, that's a carbohydrate. Galactose, maltose, sucrose, all are carbohydrates, okay? Pyran refers to a 6-membered ring containing oxygen. So a boring pyran is that. So pyranose or pyran tells you that this is in its cyclic hemiacetal form. All right, let's talk about beta. Beta refers to stereochemistry at, and at this point it would be good for us to number carbons, 1, 2, 3, 4, 5, 6. Glucose is a 6-carbon sugar. It is an example of a hexose. Hexoses are 6 carbon carbohydrates or sugars. Notice that in this pyranose form, carbon 6 is not part of the ring. It is exocyclic. Beta refers to stereochemistry at carbon 1. If up, it's beta. If down, it's alpha. I don't know what the air quotes are there, sorry, or ink quotes. They're not air quotes when they're written. Whatever, I can't stop. Beta went up, alpha went down. Or rather when drawn in this orientation. I'm going to always draw the ring sort of from this perspective with the oxygen sort of on the right-handed side. But when I draw the ring this way, it's beta when the OH is up. It's alpha when the OH is down. Yes?
So I thought it was more of an issue of cis and trans with the Ch23 group on the other side.
You can see it that way, that's fine. That actually probably works. Unless you're changing stereochemistry of that Ch2OH group.
If it was an L sugar and the Ch2OH group was going down, would it then be, would this conformation be alpha instead of
That's a
beta?
good question, and I don't know about nomenclature of L sugars. It's a good question. All right, D refers to stereochemistry at carbon 5. That is the, sorry, it's not carbon 6. I wanted to, we will call this whole thing carbon 6 in its OH group, Ch2OH. The term for that is a hydroxymethyl group. So if the hydroxymethyl group is up, it is a D sugar. If the hydroxymethyl group is down, it is an L sugar. D and L are old stereochemistry terms from a long time ago, back before there were really good ways to tell what something's absolute stereochemistry was. D and L were stereochemical terms that were assigned by analogy to a much shorter sugar called, I think, glyceraldehyde. It's a term we've kept. There is no correlation between R and S versus D and L. Trying to think what else. There's also no correlation anymore between D and L and whether it rotates light in the left-handed direction versus the right-handed direction. So it's sort of an arcane term, but D means that the hydroxymethyl group on carbon 5 is up. Yeah.
That includes carbons 5 and 6 in that group. Just carbon 6 in the OH group.
Well, the stereochemistry is at carbon 5, but the group attached to carbon 5 is carbon 6, I guess. Yeah. Okay, other questions there? Gluco refers to stereochemistry at carbons 2, 3, and 4. When drawn in this most stable chair conformation, the stereochemistry at carbons 2, 3, and 4 is all equatorial. Okay, or I guess down, up, down. Okay? You don't know that from the name, but that's now what the name means. The easiest thing I can tell you is to memorize the structure of beta-D-glucopyranose. It's easy because everything's equatorial. And then you can know the structures of the other sugars relative to beta-D-glucopyranose. For example, if I were to tell you to draw alpha-D-glucopyranose, that would be easy. You would keep everything the same. You're still going to have D stereochemistry at carbon 5. You're still going to have this down, up, down, all equatorial stereochemistry at carbons 2, 3, and 4 respectively. But at carbon 1, now your OH group will be down. Okay? All right. What's the relationship in stereochemistry between beta D-glucopyranose and alpha D-glucopyranose? Oh, that's a hellish blast from your past, right? 351. What are these? Diastereomers and enantiomers. They're diastereomers. They differ at least one, but not all stereocenters. Yes, the beta and alpha forms are diastereomers. They're a special kind of diastereomer. They differ at only one carbon. So we have two words for that. One of that is a general word that refers to diastereomers that differ only at one carbon. That is called an epimer. Alpha D-glucopyranose is the C1 epimer of beta D-glucopyranose. They differ only at carbon 1. Another term we will introduce is they are anomers of each other. And anomers refers to differing stereochemistry at a hemiacetal carbon. Carbon 1 is often called the anomeric carbon. That's the hemiacetal carbon. So you're going to hear those terms. People will call beta D-glucopyranose the beta anomer or the beta diastereomer. People will call alpha D-glucopyranose the alpha anomer or the alpha diastereomer, the alpha epimer. All of those you can use interchangeably. The anomeric carbon is the hemiacetal, or it is either, in this case, it's a hemiacetal, but in other carbohydrates it might be an acetal carbon, but it's the one that would have been an aldehyde or a ketone in an open chain form. Okay, there's 3 other carbohydrate structures I want you to know, memorize. Then we're also going to talk about structures of carbohydrates where multiple units are linked together. So yeah, stay tuned, have a good weekend, and I will see you Monday.